Today we'll be continuing from where we had to leave off abruptly due to certain technical issues I was having with my laptop. So, a quick review. We were talking about enzymes, that's chapter 5, and enzymes are biological catalysts. So, a catalyst is a substance that increases the rate of a chemical reaction and is not changed by the reaction. It brings the reactants close together for the reaction to occur. Right? Enzymes are proteins and are involved in all metabolic processes. Those could be processes in which bonds are being broken, and so larger molecules are being broken into smaller ones. Those are called catabolic reactions. An example is digestion. And then there are the types of reactions where bonds are being made. So smaller molecules are built into larger ones. The term used for that is anabolic. So those are anabolic reactions. An example is protein synthesis from amino acids, or the synthesis of DNA or RNA from nucleotide bases. So individually, those are catabolic and anabolic, and together they are termed as metabolic. Enzymes are involved in all metabolic, right? So we mentioned that. Now, the thing about enzymes is that they speed up reactions that on their own would take up ridiculously long, and the time span a single reaction would take is not conducive, it is not supportive of life. So, right, living organisms need these reactions to happen very quickly to be able to sustain life, for the nutrients to be readily available, quickly available after they have been consumed, or proteins to be synthesized as and when they are needed. They can't wait for multiple hours, let alone days, months, or years for that to happen. An example of how quick enzymes can be: catalyzes the conversion of hydrogen peroxide into water and oxygen. It is present in right throughout the body in cells, and it can convert 40,000 molecules of hydrogen peroxide per second. So that's how fast they are. Enzyme specificity is what we were talking about before class ended. So enzymes are specific as to which substrate binds to them. Again, depending on whether the reaction is anabolic or catabolic. So you have multiple reactants going to a single product, or one product being split into multiple, a single reactant, sorry, being split into multiple products. So the way the protein is folded in the enzyme, it has what's called an active site, and that is where the substrate or substrates, if there's more than one, will bind, and the shape is specific. So a very crude illustration here, right? The substrate you would expect to fit, this is the black one's the enzyme, and so substrate you would expect to fit is the one with the pointed right edge. And even though if there's something similar, right, but for example it's a rounded edge, you would not expect for that to fit. Now it's not as simple as that, but as far as the syllabus is concerned, right, we don't talk about what else happens, right, with enzymes. Similar substrates can fit in, and there are other obviously details to that, but again that is beyond the scope of the syllabus for O level. And then examples that they would give would be very different. So what they show is, for example, you've got this substrate, and then you've got, for example, uh, something like this. And so obviously this is also not what you would expect, because the shape is very different, right? Not you can't even call that similar. Then some enzymes are able to catalyze more than more than one reaction. So more than one reaction. Most enzymes catalyze, right, only one reaction. So that's where we were interrupted by my laptop. What this means is, for example, you have the starch molecule, and it is being broken down into maltose by the enzyme amylase, right? So this enzyme amylase is breaking it. It's anabolic reaction, catabolic, sorry. Sorry, I did say I confused myself. That's a catabolic reaction, and right, you're getting maltose. Now, maltose can be further broken down into glucose, and you would expect, well, starch is multiple glucose units together, and so if it's breaking one bond for the starch, right, and maltose is just two glucose units, so the same enzyme can break it. No, right? There's a different enzyme for going from maltose to glucose, and that is maltase, right? So you've got different enzymes for different steps. There are enzymes, for example, the enzyme that converts ATP to ADP, right, is called ATPase. ATP is adenosine triphosphate. ADP is adenosine diphosphate. We will come across these guys when we cover the chapter of respiration. Okay, basically ATP is what provides energy inside the cell, right? So inside living systems, it is called the uh, universal energy carrier molecule. Right, that's not the name. The name is ATP, adenosine triphosphate, but this is what it is termed as. It's dubbed as, right? Because all living organisms, the energy that they deal in is in the form of the ATP molecule. Anyway, so this is a reversible reaction. Now, in cases of reversible reactions as well, right, it's not necessary that a single enzyme goes both ways, right? It's possible that one path of the reversible reaction is catalyzed by a particular enzyme, and then the reverse path is catalyzed by a different enzyme. So, but in this case, it's an example of an enzyme which catalyzes both ways. So it converts ATP to ADP and then ADP back to ATP, right? So point being that different enzymes, different functions, some can catalyze more than one reaction, whether it's different steps of a process or reversible reactions, whereas others are really specific to a single reaction. Now, right, as far as the reason for that is the active site. So the active site of an enzyme is complementary to the substrate, right? Complementary meaning specific. So the shape, as we talked about, right? The shape of the active site of the enzyme will, only allow us, a particular substrate of that shape to fit in and no other. And when that happens, so when you have the enzyme and the substrate bindings, if I just draw this, and then I say, right, this is the substrate that's bound to it, right, taken from here. So the enzyme plus the substrate is bound. And so what we are seeing over here is the formation of an enzyme substrate complex. And the binding is termed as the lock and key hypothesis, right? The way it binds, the method of its binding is termed as a lock and key hypothesis, where the enzyme is considered as the lock and the substrate is considered as the key, right? And again, so we know that keys and locks are specific, they're complementary as the term is used, right? So you can't take any key and fit it into any lock and expect the lock to open. No. So similarly, right, the enzyme is the lock, the substrate is the key, and right, only the correct substrate will be able to fit into the corresponding, the appropriate enzyme, and then the reaction will occur. So, right, let's say for the sake of, this is maltase and this is maltose, right? And so what you're going to get now is, as you know, right, the enzyme is not going to change. So that's still the enzyme. And what you're going to get now, for example, maybe is the substrate broken into two. So maybe just for the sake of illustration, again, you still have the enzyme, maltase, and these are two glucose molecules. All right. I mean, the thing is that, right, in a cell, there are many enzymes present at the same time within the cell, and there are enzymes that are also released outside the cell, right? So, catalase, as we mentioned, is what's called an intracellular enzyme. Why? Right? Intra means inside. So this one's found inside the cell. And then there are other enzymes such as maltase, which are extracellular. Extracellular meaning that they are secreted outside the cell. Why are they secreted outside the cell? So maltase is what you would find in the intestine, right? And so the food that's coming in from the rest of the alimentary canal and reaching the intestine, that's obviously not inside the cell, that's in the lumen of the intestine, the space of the intestine. And so the enzymes are produced by the cells of the intestine, and then they are, right, released into the cavity of the intestine, the space of the intestine where the food is. And that's where the digestion occurs. So why am I mentioning this? To answer your question, there are many enzymes and many substrates present, right? An enzyme is usually only produced when it is needed, right? So if you don't have something to work on, then you probably won't have the enzyme produced either, because making an enzyme from amino acids is utilizing the amino acids, so it's utilizing resources that the body needs, for example, to make other proteins. And so if you don't need an enzyme, you won't use those amino acids because the enzyme will be useless, and so you are wasting resources. Of course, none of this is under your voluntary control, right? The body is managing itself. So if there is a substrate to be digested that the body can process, right, whether it's catabolic or anabolic, right, if there is something to happen, then the body will produce the relevant enzyme. So in most cases, it's not that the substrate finds the enzyme, it's, normally the enzyme is released into the space where the substrate is. Okay, does that answer your question? Okay. And the way the enzyme substrate complex is formed is, you've got the enzyme over here, and again, I'm not going to bother. So you've got the substrate over here, and it does happen due to collisions, So it's sort of random collision, but the more the substrate molecules, the easier it will be for them to bind to the enzyme, right? And then we'll just talk about temperature and how that will affect the rate of enzyme activity. Okay, what else? So, all right. Now, effects of temperature and pH. Starting with temperature. So in general, what happens with temperature is when you increase a temperature, increase temperature, uh, right? You increase the kinetic energy of particles. Okay, and when the kinetic energy is increased, you increase the number of collisions of particles. And in case of enzymes, it's not only the number of collisions, which sounds random, you're also increasing the number of effective collisions. When you talk about effective collisions, we are talking about the enzyme and the substrate form they come into the right position and they form the enzyme substrate complex. And when that is happening, right, you are having more of that particular reaction taking place, and so you are increasing the rate of enzyme activity. Okay. Now, in general, for every 10 degrees centigrade rise in temperature, the enzyme activity is doubled. So we could say, oh, well, we keep increasing the temperature and enzyme activity will keep increasing, so that's good. No. Why? Because the thing is that enzymes are proteins, and proteins are sensitive to heat, right? Which means there's only a certain temperature up till which they will be able to work properly, right? If the temperature goes too high, the protein, any protein, in fact, not just enzyme, right? Any protein, even if it's not an enzyme, if it's a protein hormone, if it's a protein that's in the cell surface membrane, if it's a protein somewhere else in some other cell in some other part of the body, if it's a neurotransmitter protein, whatever, right? If the temperature is too high, the protein will undergo denaturation. It will become denatured, which means that due to the heat, its shape will change permanently, right? So even like if the temperature goes too high, right, the shape So even if you decrease the temperature, the temperature is decreased after that, right? It will not, recover. Okay. So with that, what we have, because the shape has changed, it's just like it's, and the reason why the shape changes, right, permanently, is because of the intermolecular bonds. The temperature affects the, this particular detail is not given in your text, it's not given in the syllabus, but temperature affects the intermolecular, right, bonds. And so that's why, so this is just like put an asterisk on that, just for the sake of understanding. So what do we get? Well, if we study the effect of temperature against, enzyme activity, I'll just come to that. And so over here you got temperature in degrees centigrade, and over here you have enzyme activity. And so sort of what you see, and I'm just making like a generalized, right, curve over here. Looks something like this. And so you see that there's a particular temperature where the enzyme activity is the highest, right? So this is called the optimum temperature for that particular enzyme. Why? Because the enzyme activity, the rate of enzyme activity is highest. Now, in living organisms and in humans in particular, right, the optimum temperature for enzymes is about 40 degrees centigrade, right? The core human body temperature is about 37 degrees centigrade, right? So very close to the optimum. And so, and when you're fine and healthy, you would expect, you know, the enzymes in the body to be working at near optimum, right? If, right, the temperature of the body rises, then, if it stays like that for too long, then it can damage the enzymes, and that would cause a decrease, or right. So in the body, normally you don't go to very high temperatures. That's why body temperature is measured in degrees Fahrenheit. So when you see, oh, it's 98.6 or whatever, right? So that's normal body temperature. If it's going to 99, then you say, okay, you're slightly feverish. 100, 101, right? When it's getting to 102, 103, right? Then it becomes dangerous. Why? Because those prolonged, and this is, we're talking about obviously degrees Fahrenheit, not degrees centigrade, right? Because at 103 degrees centigrade, you will definitely be cooked. So right, as the temperature rises, the one reason why it's dangerous is because that, right, you need to take a cold shower or do something to bring your body temperature down, because if you stay in a state of fever for too long and a high fever, then that will end up damaging the proteins in your body. And then if the proteins are damaged, then the activity will be reduced. And if the activity is reduced, then the metabolic processes and all the other functions like the proteins in the cell membrane or the neurotransmitters or whatever they are, right? They will not work properly, and so your entire body will start, having feeling that negative effect. Now, as you see in the curve, right, when the temperature is rising above optimum, the enzyme activity is actually falling, and that's for the same reason I mentioned, right, is that the protein is becoming denatured, the shape is changing. So, just a sec, just a sec. So, what happens is the reason why it's not immediately going to zero is because as the temperature increases above optimum, right, not all the enzymes, and right, we're talking about enzymes over here, but again, it happens to proteins in general, are denatured immediately. So some will start losing their shape, others will not as quickly, and so you see a fall in enzyme activity, right? And as you keep increasing temperature, more and more of those enzyme molecules will become denatured until at one point it will be so low and it will actually go to zero when all the enzymes are denatured. So you have lost all of that, and so obviously no, you will not have any activity. On the flip side, right, if you decrease the temperature, so right, that's going to reduce the kinetic energy, and so reducing the temperature is not going to cause denaturation, right? It's going to reduce the kinetic energy, and so that's going to reduce the number of collisions and effective collisions. Right, and to, the lower you go, because you're taking kinetic energy out of the system, and so there won't be enough effective collisions taking place for a very good rate of enzyme activity. So you know the efficiency of the enzyme will decrease. Does this make sense? Okay. So that's about, right, temperature and enzyme. Scientists, right, so this is about the enzymes in the body, right? Now different organisms have different temperatures, uh, right? Living organisms, animals, mammals, vertebrates, all of those, right, they have more or less the same working range perhaps, right? Some can, the ones that live in like really cold water and uh, have enzymes, and especially fish, right? So fish are cold-blooded and they live in cold water. So we can assume that their enzymes are suited to work in those cold temperatures, right? Our enzymes are not. So, right, the optimum pH for the enzymes of different organisms is different. And you can have enzymes with optimum temperatures that are low. You can have other organisms with optimum temperatures that can go up till 80, 85 degrees centigrade. So there are bacteria that have been found in hot springs, right, and thermal vents under sea where the temperature is like 90 plus, and they've found bacteria living over there, and so if they're living, that means their enzymes are working at that high temperature. Okay, next up we have time is, right, pH. Now even within the body, right, different enzymes have different optimum pH. Now, I don't need to explain that. You understand the concept of optimum. So, as with optimum temperature, where you have maximum rate of enzyme activity, right? And right, the same goes with pH. So, if you plot that, try and make a straight line, acceptable, and so you would notice the same for right. The range for pH is much smaller, right? So you can have something like this. You can have something, right? So these are all different enzymes, and actually copying the graph that's made in the book. So you've got rate over here, you've got pH over here, and so you're starting from 0 all the way till 14, which is the pH scale. And so, for example, what you have over here is pepsin, which is what you would find in your stomach to digest proteins. And so it has an optimum pH of 2, right? And these guys are probably, perhaps intestinal enzymes, right, which need an alkaline pH. So maybe pH uh, 8 or 9, right? So just as a general, right, the pH in your mouth and the intestine is alkaline, and the pH in the stomach is acidic, right? The pH in cells, inside the cells, is neutral. So as I mentioned, right, the digestive enzymes that are involved in digestion of food, they are released outside the cells. So the stomach cavity where food is, that is acidic. The mouth cavity and the cavity of the intestine, that is alkaline. But if there's any enzyme activity happening inside the cell, the pH of the cell is neutral, right? So obviously the enzymes you find in these places are suited to work best at those pH ranges. So when the amylase that is present in your saliva, which is right an alkaline environment, it moves to the stomach, that's acidic, amylase will stop working, right? Why? Because it cannot function in acidic acidic environment. And when the contents of your stomach from move to the intestine, and so they're moving from an acidic environment to an alkaline environment, and so bile and other things are added to neutralize the acid, and so pepsin will not work in the intestine because it needs an acidic, very acidic environment. In fact, right, the pH is 2 in the stomach, and it's alkaline in the intestine, and so pepsin will not work, and so there are other enzymes in the intestine that will work at an alkaline pH. Okay. Now the thing about pH is it's not as drastic as uh, temperature, right? So changes in pH affect enzyme activity, obviously, but right, it is recoverable. Meaning what? Right? If the change is not too drastic, if the change isn't too drastic, right? So if pepsin works at pH 2 and you take it to pH 3, 4, 5 maybe, you will lose enzyme activity. But then if you take it back to pH 2, it will start working again, right? But if the change is too drastic, as happens in the body, that from the stomach is going to the intestine, so it's going from pH 2 to pH 8 and 9, then you're going to lose, because again it's going to affect the bonds within the protein molecule and it's going to become denatured, right? The book says The book says that it is reversible, right? So the effect of temperature changes, especially increasing temperature, is not reversible, but the effect of changes of pH are reversible. So as far as your O-level syllabus is concerned, you stick with that, right? That if the pH changes, then you lose enzyme activity, but if you go back to its optimum, then you can regain enzyme activity. Power of hydrogen. Okay, and I believe that brings us to the end of this particular chapter. How we can reverse the pH? I mean, you can't do it. Yeah, go ahead. Well, again, right, the details are not part of your syllabus. The only thing that your syllabus mentions is that you can regain enzyme activity if you go back to its working pH, right? How that happens, why that happens, again, right? So for that, you will have to take a look at the how the protein molecules are folded, what are the bonds within the protein molecules, the interactions between the different amino acids, etc., and how those are affected by temperature or pH. And that, if you decide to take biology in A level, then I will be gladly, I'll explain it. But it is beyond the scope of your syllabus in O level. You can, of course, research it on your own, right, if you're interested. So definitely, right. I do encourage curiosity and inquisitiveness, so that's a good thing. Okay, so I mean, I had planned on completing this chapter yesterday. Of course, that didn't happen. Now we have about 10 minutes of class, but I don't know, right? I think, how about we just start the next chapter next time? Is that okay? So yeah, that's all for today, right? So next time, obviously, we'll be moving on to chapter 6, which is plant nutrition. Okay? All right then. Have a good weekend. Allah.